Zoom recording. Okay. Hello, everyone. This is the middle school USAVO club meeting. Today, we're going to be going over Campbell's chapters 14, 15, and 16. Let's get right into it. We're going to start with chapter 14 regarding Mendel and the gene idea. Mendelian genetics assumes that each gene has two alleles, meaning two different versions of DNA for that gene. And a classical Mendelian genetics assumes that an allele is either dominant or recessive. We'll get into what that means in a second, but you should know these definitions. A homozygote is an organism that has two identical alleles for a gene. Remember how all diploid organisms like us, we have two sets of chromosomes, one from our mom, one from our dad. And so there are every chromosome has one allele, one locus for the gene. So an organism with two identical alleles on both of the chromosomes, the same allele on both chromosomes, is a homozygote. On the other hand, if an organism has different alleles on the two chromosomes, then they are a heterozygote. Then phenotype versus genotype. Phenotype oh my God, it's Derek. Phenotype is the actual characteristics of the organism, what they look like, maybe some other intrinsic stuff. Genotype is the genetic makeup. So the genotype determines the phenotype, but these two things are not the same. So for example, whether or not an organism is a homozygote or a heterozygote, that's the genotype. But the phenotype would be something physical that you can see, that you can detect, like eye color. And more vocab, test cross in Mendelian. Well, I'll just show you in a bit. Laws of inheritance. Mendel worked with pea plants, and he determined that they have variations in character. Here's another definition: true breeding individuals. This is where these are organisms that these are like a group of organisms that if you repetitively interbreed them with each other, they will keep producing the same phenotype, meaning that, like, for example, if you have a purple pea plant, after a group of true breeding purple pea plants, after years of breeding and breeding and breeding between each pea plant, the final one is the pea plants, the offspring are still going to be purple because that is the definition of true breeding. I don't think we have time for a Kahoot. More definitions. The P generation, or the parent generation, is the first group of organisms that you're working with, the first group that you interbreed. Then the offspring of the P generation is called the F1, then the F2, and so on, F3, F4, blah, 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 blah. If anyone has any questions at any point, please just type in chat. Laws of inheritance. So Mendel came up with a few like theories, laws that governs how genetics works. One of which is the law of segregation. This has to do with organisms having two sets of chromosomes, right? When I'm getting mixed up. Each parent will only pass one allele to the offspring. Because each parent only passes on one chromosome, that chromosome can only contain a single allele. And what that means is the parent will only contribute half of the offspring's genotype. This is where we get into the dominant versus recessive allele. This is like for classical Mendelian genetics. The dominant allele codes for a protein. It does a specific function. So if you have a dominant allele, then the dominant allele will determine your phenotype. The recessive allele, on the other hand, is characterized by a lack of function. It doesn't, in this case, if you're talking about Mendelian genetics, a recessive allele does nothing. But if you have two recessive alleles, that gene will do nothing as a whole, and therefore that will lead to a phenotype. Does that make sense? Right. The two alleles for a trait separate during meiosis. In gamete formation, each gamete contains one chromosome from the parent, and therefore can only contain one allele for a given gene. And in the bottom right corner, I'm not sure why it's so small, but hopefully you've seen a Punnett square before. It is used to determine, you take the genotypes of the parents, and then you determine the possible genotypes and phenotypes of the offspring. Yeah. Inheritance. So, like I tried to emphasize the importance of the things that we just discussed, the dominant and recessive alleles. They only apply to Mendelian genetics because Mendelian genetics is complete dominance. However, there are at least there are two more types of inheritance that a gene could be. The first one that we just talked about is complete dominance. So that means that the dominant allele, the one that codes for a functioning protein, will always dictate the phenotype. So for example, if you have a flower with a gene that codes for flower color, right? And that gene can either be red or white. Red is dominant to white. If the inheritance is completely dominant and you have a heterozygote individual, heterozygote, remember, is a organism that has two different alleles. If you have a heterozygote individual, you have a red and a white allele, their flower will be red because the dominant gene, the red gene, the red allele overpowers the white allele, which is recessive. However, the second type of inheritance is incomplete dominance. This is like a blending of the alleles. It's not so much. Is that why I saw pink flower? Can you explain your question a bit more? I'm not sure I understand.
Like there's like a breed of flowers that were red and that like two different colors, red and white, and I saw pink flowers in between them. Is this why they're pink flowers?
If you have a flower that is both red and white, that is codominance. If you
No,
have
okay.
get back to me, like send me a DM or something, or we can talk about it during break if we have a break. Back to this. Incomplete dominance is when you have, let's say yes. What do you mean yes? Incomplete dominance is when you have a heterozygote individual with a red allele and a white allele. They combine, you blend the traits, and you see a pink flower. In this case, there's both code for proteins, both have functions, and as a result, the offspring will look like a mix of the two parents. Neither allele is completely dominant over the other. The third common method of inheritance is codominance, where both phenotypes, both genes are expressed equally. So in this case, if you have a flower with a red and white allele, you're not going to see only red petals, you're not going to see pink petals. What you're going to see instead is some petals are red and some petals are white. So you're going to have a flower with both red and white petals. You should keep in mind that blood type is codominant. Blood type has three alleles, allele for A, allele for B, and an allele for O. Just remember that blood type is codominant. Inheritance patterns of non-Mendelian genetics. So these are it's little eyes. Oh my god. I was talking to Derek about blood type. Back to this. There are non-Mendelian genetics. These are more examples of how the Mendelian rule of two different alleles, one dominant over the other, is not always true. One example of this is pleiotropy, where a gene has multiple phenotypic effects. What does this mean? This means that a single gene will code for a protein product that affects many things. Pleiotropy. Um, a single gene will code for a protein that has many different effects. So like a lot of these pleiotropic genes are regulator genes that determine whether or not a gene is expressed, whether or not other genes are expressed. A second example is epistasis. Epistasis is where the expression of one gene at one locus affects the expression of another gene at a different locus. In the bottom right here, you see a cross between two black Labradors, right? In this case, we're talking about two different genes. One determines coat color. The B, it determines coat color. The the E determines whether or not the Labrador produces pigment. So as you can see here, if the gene that produces pigment is recessive, then the Labrador will have no pigment in its coat, which results in a yellowish phenotype. Whereas if it does produce pigment, then it will create some like black or brown hybrids, black or brown phenotypes. This is an example of epistasis because the gene that codes for pigment production will affect the coat color of the Labradors, which is a separate gene, right? Oh, I clicked that. Polygenic inheritance, traits vary on a gradient. So this is polygenic inheritance just means that one specific phenotypic trait is determined by many different genes. Like for example, skin color is not determined by one gene. It's determined by a lot of combination of genes. So that's why there are so many. There's a gradient of skin colors. As you can see here, this is like a very simplified version of the genes that code for skin color. You can see how there are many combinations of these just three alleles leading to a gradient of colors like as you go down and across, right? Okay. Inheritance patterns, nature versus nurture. Another exception to Mendelian genetics is that not everything is determined by your genotype. Your phenotype is also influenced by your environment. So in this case, for example, hydrangeas have a specific gene coding for flower color, but depending on what kind of environment they're grown in, they can develop very radically different flower colors. In this case, if there's a hydrangea grown in very acidic soil with lots of free aluminum, then the flowers will be blue. If the hydrangeas are grown in basic soil, they'll be red. These kinds of genes are called multifactorial genes, which the gene is affected by multiple factors, multifactorial, not the math thing, the multiple factors. Human inheritance. We have two different types of chromosomes, broadly speaking. We have autosomes, which are like very big chromosomes. They code for proteins that are very important in function. And then we have the sex chromosomes, the X and Y, which their main job is to ensure function of reproductive systems and determine the offspring's sex. And when we talk about inheritance, we generally think of recessive and dominant alleles. A gene can either be autosomal recessive, autosomal dominant, X-linked recessive, X-linked dominant. The first part tells you where the gene is. If it says autosomal, it means the gene is on an autosome. Same for X-linked. And then dominant versus recessive tells you the type of gene. Human, and then here we can see, well, this is what a pedigree looks like. It's a pedigree shows you the ancestry of, like, it's like a family tree, but it also shows you whether or not individuals are affected by this condition and like how the generations work. So here's how you read a pedigree, right? If it's a circle, then it's a female. Like in this case, this circle, actually no, this circle, female. This squares are males, right? And if there's a line drawn between a circle and a square, that means that they're married, they had children, and the line drawn down, these three are the children of 1 and 2. See, they connect, they draw down, and then the direct descendants. If the shape is black, it's colored in, that means that that individual is affected by whatever condition you're studying. If the square is white, or if the shape is white, sorry, that means that that individual is unaffected. So is this individual number 7? Tell me a little bit about them. What's their biological sex? Are they affected by this condition? Please DM me in chat. You guys have 15 seconds. Number 7 right here. Oh, Angela. Oh my god. Okay. Alright. So why did I say it? Sorry. One thing I want to point out is whether or not the circle is shaded does not tell you whether the condition is recessive or dominant. Recessive or dominant, that is the someone answered incorrectly. That's why I'm talking about it, Derek. That is why no, sorry, I lost my train of thought. The shading of the shapes does not tell you whether or not the condition is recessive or dominant. That will usually be given to you, or you'll have to deduce that. In this case, it's a circle, it's a female, and then it's not shaded in, so it's unaffected. Okay, and this is autosomal recessive. That's not important for now. Autosomal dominant. It's the same thing for this. Shaded in is affected, unshaded is unaffected. One thing I want to point out is in the autosomal recessive, there's a lot more unaffected individuals in the 2nd generation, right? In the F1 generation, but in the autosomal dominant version of a gene, there are a lot more affected 2nd generation individuals because wherever this autosomal dominant allele is present, the person will have that condition. I think I need to move a bit faster. This is X-linked dominant. There's not really much to say here. One thing to point out is males only have 1 X chromosome, and that I'll show you why that's important shortly. There's not much to say about this slide. And here, X-linked recessive. So what does X-linked recessive mean? That means that the gene is located on the X chromosome, and it's you need 2 copies of an X-linked recessive gene to gain the condition. Actually, all of your X chromosomes need to express a recessive gene to have this condition, right? That's remember when I mentioned before, males only have 1 X chromosome. That means that if they have a single X-linked recessive X-linked recessive gene, then they will be affected by this condition. As you can see here, there are 5 affected males and only 3 affected females. Generally, if you see X-linked recessive, you should think differences in sex, because if it's X-linked recessive, the males will be affected more. Human inheritance, these are some recessive disorders. Recessive disorders, you can have carriers, which it just means it's a carrier is a person who carries a recessive allele, but is not affected by the condition because they have another dominant allele. So they can keep that gene in the gene pool without having negative effects to themselves. In this case, only homozygous recessive individuals, so individuals with 2 recessive alleles, will be phenotypically affected. Here are some examples of recessive disorders. Cystic fibrosis, you get edema, lots of mucus. Albinism, you don't produce melanin, no pigments. So you kind of your skin is pale, your eyes are pink. Sickle cell disease. This is an important one. You guys should know this. It's when a single mutation causes blood cells to form spindles. Normally they're nice and round, but if you have sickle cell disease, if you have the disease, that means you have 2 recessive alleles. And if you have the disease, whenever oxygen concentration goes down, the blood cells will form little spindle shapes. They're like curved things. Yeah, like a pointy banana. And this will cause blood cells to clog in your capillaries. So it'll cause a lot of internal damage. That's very bad. However, the reason that this disease, this allele is propagated throughout generations is because there's a heterozygote advantage. This allele originated in Africa, where malaria is very common. And if you're a heterozygote, you get resistance to malaria. So it's selectively, you don't want the homozygous, but you do want heterozygous. That's why there is still this allele is still present in the gene. Here are some human dominant disorders. Anyone with this gene, with a dominant gene, will be affected because of the nature of a dominant allele. These are generally less common because they can't hide in a carrier. They can't. If it's affected, then there will be negative effects. One example that you should know is Huntington's disease. It's a neurodegenerative disorder. You lose control of your muscles. The prognosis is around 40 years of age.
Wait,
Yeah, go ahead.
Another question. Do disorders ever get affected by other things what's it called again?
Like the environment or other genes?
Yeah, the environment they're living in, exposure.
Yeah.
Do they get affected by the exposure too?
Genetic diseases. Well, one disease that has a genetic component, schizophrenia, right? I think the number was 40. If you have identical twin, let's say you have a pair of identical twins, one twin develops schizophrenia. The odds of the second twin developing schizophrenia is 47%, which means that there is a strong genetic component, but it's also affected by your environment. So yeah, to answer your question, yes. And the environment does affect disorders. And these disorders can be tested when a child is still in utero, right? To get a detailed genetic makeup, you need to do procedures called invasive procedures, right? These are either amniocentesis or chorionic villus sampling. Amniocentesis is you take is you have a needle guided by ultrasound, and you collect some amniotic fluid. Then you karyotype you like determine the genes of the baby through cells floating in amniotic fluid. Chorionic villus sampling, you take a piece of the placenta and you get the baby's DNA that way. These are generally, they have risks for the fetus. And usually this is like, if you see there are risk factors for genetic disorders, you might consider doing these invasive procedures. Otherwise, generally, patients just undergo screening, like ultrasounds. You know, like the really grainy picture of black and white picture of a baby. Yeah. Now we're going. Oh, I gotta speed up. Campbell's 15, chromosomal basis of inheritance. These are some sex-linked genes. In humans, our sex is determined by an X and a Y chromosome. We have the XY system, which means that if you have a Y chromosome, you're a male, and the males will determine the sex of the offspring, right? Other animals don't. They're not every animal has the XY system. For example, grasshoppers have the X0 system, which just means if you have an even number of X chromosomes, you're a female. If you have an odd number, you're a male, right? Chickens have the ZW system. This just means it's the XY system. It's like the same thing as us, except the female determines the sex of the offspring. Females have the W chromosome if and the female determines the sex of the offspring. Yeah. And the haplodiploid system, honeybees have this, bumblebees. If you're diploid, you're a female. If you're haploid, you're a male. You don't have to remember exactly what these are. Just know that there are other ways of determining sex. Genes located on sex chromosomes are sex-linked. For humans, these are mostly X-linked because the Y chromosome is very small. Most of the genes there only code for development of reproductive system. So sex-linked genes are usually X-linked. Sex-linked genes, inheritance. Mothers will always give an X chromosome to the offspring, but fathers can either give an X or a Y. That determines what the sex of the offspring. Sex chromosomes. Each sex chromosome assorts independently into gametes. This just means that in meiosis, when gametes are formed, the chromosomes line up, they're randomly separated into one of 4 gametes. So what your genetic makeup is random, basically. That's what this means. Color blindness. Okay, this is an example of an X-linked recessive disorder. Color blindness is X-linked recessive, which means that if a daughter has color blindness, they must have 2 recessive alleles. Moreover, the daughter will get one X allele from their mom and one from their dad. Because their dad only has 1 X chromosome, and that X chromosome must be recessive, the dad must be colorblind. The mother can either be normal vision or it can either be a carrier or also affected because she has an extra X chromosome that can determine whether or not she has an extra X chromosome that determines whether or not she actually is colorblind. These are colorblindness, Duchenne, muscular dystrophy, hemophilia. These are examples of X-linked recessive disorders. X activation. So females have 2 X chromosomes, right? But you only want to have production of proteins from 1 X chromosome. If you have an extra X chromosome that's being transcribed, that just means the genes are being turned into proteins. That's, as we'll see later, that's bad for the female. So in early embryonic development, 1 X chromosome is randomly turned into a bar body. what this means is like not non-coding RNAs come in and they condense the X chromosome in a way that prevents it from being transcribed. Yeah, just know that one X chromosome is inactivated. You know what's interesting? This is not which X chromosome is inactivated is determined very early in embryonic development, but it's determined when there are like when there's more than one cell, like in the early blastula gastrula stages. One of these X chromosomes is inactivated. However, as those cells divide, the cell remembers which X chromosome has been inactivated. So the embryonic cell and all of its descendants will have the same X inactivation pattern. However, these between different like embryonic cells, the X chromosome that is inactivated can be different. So that results in like patchy phenotypes in females. Like for example, tortoiseshell cats with patches of different colored fur. The different colors are the results of different X inactivation in the embryonic stage. Now we get to the concept of linked genes. A gene is linked when the two genes are linked when they're very close together on a chromosome. This is the case because in meiosis, there's a process called crossing over, which means different chromosomes, homologous chromosomes will exchange segments of like DNA, so they'll trade pieces of themselves with each other, and that results in more genetic variation. However, if two genes are very close together, they'll often if a segment of a chromosome is going to be exchanged with another, they'll often be traded together. Does that make sense? Because they're very close together, there's not much space between them, and it's hard to cut the what's up, Aspen?
If you had two albinos like that mated with each other, would you get an albino?
If you have two albinos, oh, let's go back. Where is it? Albinism. Oh, I missed it. I'm kind of done. If you had two albinisms that mated, well, albinism is X-linked recessive. But if wait, no, it's just recessive. That means that both parents have two copies of a recessive allele, which means that no matter what kind of like randomness happens, two recessive alleles will be passed to the offspring, which will result in an albino child. So yes, that was a very long-winded explanation. But yeah, to answer the question, yeah. You want to lower your hand, or do you have another question? I'm gonna move on. Where was I? Linked genes. Yeah, so if two genes are close together, and it's hard to separate the two, it's hard to trade the two genes without it's hard to trade the two genes it's hard to trade only one of the genes because you have to like cut the distance between the two genes very precisely. And that generally means that the two alleles for a gene, two different alleles for a gene are associated with each other. And this just shows that sometimes like if there are linked genes, then the numbers of phenotypes that you have are going to be much less. No recombination occurs, only two potential gamete types instead of four. Although it's less likely, recombination you can still split the alleles, it's just not as likely. And a recombinant offspring is an offspring where it defies the it defies the linked genes. In this case, the dominant alleles for color and P shape are linked together. The recessive ones are linked together. So if you have an allele, if you have a gamete that has a dominant and a recessive allele, then that's a recombinant offspring. Yeah. Any questions? Alteration of chromosome number. This happens because of an error in meiosis. When the gametes form, sometimes there are errors in their replication. This is generally very bad for the organism. Abnormal chromosome numbers could be caused by non-disjunction. Homologous chromosomes, when they line up for during meiosis, sometimes they don't separate properly, and more chromosomes go to one side than to the other. Incorrect number of chromosomes is called aneuploidy. If you're lacking one chromosome, you're monosomic. If you have an extra chromosome, you're trisomic. You have 3 chromosomes, 1 chromosome. Aneuploidy just means you have an incorrect number of chromosomes. Trisomy 21, notably, is Down syndrome. Certain organisms can have more than 2 complete chromosome sets. So polyploidy is not the same as aneuploidy. Aneuploidy means you're still like roughly diploid, but you just have a duplication, or you're missing 1 chromosome. Polyploidy, on the other hand, is you duplicate your entire genome. Like, let's say us humans, a polyploid human would have 92 alleles, whereas an aneuploid human, we usually have like 40, 45 or 47, like 1 more or 1 less. Alterations of chromosome structure. This can happen either deletion, duplication, inversion, translocation. These are some kinds of mutations that can happen to like large segments of DNA. This happens mostly during mitosis and meiosis. Deletion, it's self-explanatory. You remove a piece of a chromosome. Duplication, you double the amount of genes. This generally happens when DNA is being replicated, and the polymerase, the thing that makes the DNA will fall off, and then it will go back to the start of the gene and like make another copy. Yeah, the polymerase slips. Inversion, gene may be attached in the reverse order. Sometimes when DNA is being not repaired, if you're exchanging homologous DNA, sometimes when you reattach the gene, the gene may be attached in the reverse order. Translocation, gene is moved to another section of the chromosome. This could be pretty bad because the location of the chromosome, the location of the gene in your nucleus is important. Yeah, I think we will get into that in a bit. Human disorders due to chromosome alteration. First one, notably, like I said, Down syndrome is trisomy 21. People with Down syndrome, they have unique facial features, and some mental retardation, but generally many lead like normal, somewhat like not normal, but their lives aren't that different, and they're contributing members to society. What was I gonna say? Oh, a general rule for aneuploidy: the lower the chromosome number, the more deadly an aneuploid of that allele of that chromosome is. So a duplication of chromosome 1 would be the deadliest. This is because chromosome 1 is the biggest and it controls the most number of genes. Yeah, so chromosome 21 is near like the lower, like the higher end of chromosome number. Therefore, it's less dangerous, it's less effective. Less dangerous. Aneuploidy of sex chromosomes. Klinefelter syndrome. This is where a guy has an extra X chromosome. You have they have XXY. The phenotypic effects of this disorder is you're usually taller, more thin, and you might, so I think some people with Klinefelters have an underdeveloped reproductive system. Actual Y chromosome, it's not that big of a deal because again, the Y chromosome is very small. Trisomy X, I forgot to read up on this. I don't think this one is that important either. But Turner syndrome, these are females with only one X chromosome. Generally, because they're lacking an X chromosome, they will have underdeveloped reproductive systems, though most of them will be sterile. But other than that, I'm not sure if there are other major phenotypic effects. Structurally altered chromosome. These are two that you should like maybe keep in the back of your mind, like know their names. Prelude shot. This is a deletion in chromosome 5, not of chromosome 5, but deletion in chromosome 5. Oh, thank you, Angela. That is what trisomy X can lead to. This is often like an infant. It kills you when you're an infant. And your mental processes don't develop properly, and notably when the infant cries, it sounds like the crying of a cat. Another thing, Philadelphia chromosome leads to CML, chronic myelogenous leukemia. It's an exchange between two non-homologous chromosomes, chromosome 22 and chromosome 9. They like switch, and the change in location leads to leukemia, which is a blood cancer. Exceptions to inheritance, genomic imprinting. So this is as you live, the choices that you make every day affect your DNA. This is by adding methyl groups or acetyl groups to like DNA or histones that will make certain parts of DNA more transcribable or less transcribable. This is an example of the environment affecting like your genes and therefore the phenotype. Yeah. Oh, wait, no, I was thinking of something else. In this case, genomic imprinting is actually never mind. Or you also have organelle genes, extranuclear genes. They are inherited from outside the nucleus. These are like mitochondrial genes. Mitochondria evolved from endosymbionts, so they still have like circular bits of DNA. Some disorders are carried through mitochondrial genes. One thing you should know is that these organelle genes are only passed on through the mother. The mother contributes all of the extranuclear genes of an egg, of a zygote. One disease that's caused by mitochondrial genes, ragged red fiber. Yeah. Campbell 16, the molecular basis of inheritance. Okay. Here, Hershey and Chase, they used radioactive sulfur and phosphorus to determine that DNA was a hereditary material. Basically, they just showed that they introduced radioactive elements into the cell. Sulfur would be incorporated into proteins. Phosphorus would be incorporated into DNA. If DNA was the hereditary material, then the phosphorus would be seen in later generations because DNA is replicated. If proteins were the genetic material, then same for sulfur. What they found is that DNA is in fact the hereditary material. Bacteria can undergo transformation where you take bacteria can, like, if there is a plasmid or a section of DNA in solution in their environment, they can absorb it and incorporate it into their DNA. That's another way that they can increase their genetic variability. This is one important reason, one important thing about this is if you have a resistance plasmid or a virulent plasmid, these are plasmids that can affect the phenotype of a bacteria that makes them dangerous to humans. If there's a lot of these plasmids, a harmless bacteria can take up one of them and then become dangerous to humans. And then Chargaff's rule is the rule of base pairing, where adenine always pairs with thymine, cytosine always pairs with guanine. Therefore, the percent of adenine in DNA will equal the percent of thymine. Yeah, and then same for cytosine and guanine. Yep. Here's a structure of DNA. Rosalind Franklin's photo 51. This was made by officially Watson and Crick discovered the structure of DNA, but they couldn't have done it without Franklin's confirmation. But they didn't credit her, which was a kind of a big move, honestly. But they constructed the anti-parallel model, which basically states that DNA has two strands, and it has a front and a back, a 5' and a 3' end, and the 5' end of one piece of one DNA matches up to the 3' end of the other DNA. So basically, the front of one piece of DNA is paired to the back of the other piece of DNA. DNA is a polymer made of nucleotides. A nucleotide consists of a phosphate group, which is a 3', a 5-carbon sugar, and deoxyribose, and a nitrogenous base. Nitrogenous bases between two pieces of DNA connect through hydrogen bonding, purines. So, and there are two types of nucleotides. There's the purines, which are big ones. They're like they have 2 rings. They're twice the size of pyrimidines. And pyrimidines, which are smaller. Adenine and guanine are purines. Cytosine and thymine are pyrimidines. A and G are purines. C and T are pyrimidines. DNA replication follows a semi-conservative model. This means that DNA will when DNA is replicated, each new strand of DNA contains one old strand and one new strand. The old DNA, like the parent DNA, is split, and new strands are synthesized along it. Yeah. Here's how DNA replication works. First, helicases, the replication assembly gathers at an origin of replication. This is just a starting point for DNA replication. In eukaryotes, there are many. In bacteria, there is a single. At these origins, replication starts and it goes both ways, and it forms like a bubble. The DNA unzips, and it opens up, and then daughter strands are synthesized along the DNA. This light blue is a daughter strand. The dark blue is a parent strand. And as the DNA keeps on replicating, the bubble expands until they meet each other and they become two long daughter DNA molecules. So what's the molecular mechanism for this? So at the replication forks, you start off with a protein called helicase. Helicase will unzip this DNA. It'll attack the origin of replication and pull it apart. It's at the front right here, and as it moves in the direction, it will continuously pull the DNA apart. Then come single-stranded binding proteins, which will attach the DNA and prevent the DNA from snapping back together. Once it's unzipped, you don't want it to come back together because then you can't replicate it. So these proteins ensure that that these strands can't come back together. Finally, there's topoisomerase, where helicase will continuously like twist the DNA to unravel it. And that might cause supercoiling, which just means the DNA, the nucleotides in front of helicase, like right here, they will get very like tangled, and sometimes if they're too tangled, they'll snap. Topolysomerase's job is to cut out some stressed pieces of DNA and cut them out, put them back in a way that relieves that stress so that the DNA molecules don't snap. Yeah. Next, our RNA polymerase will add once the DNA is opened up, RNA polymerase will add a primer. This is like a start signal for DNA polymerase. Polymerase is a protein that lays down nucleotides. In this case, RNA polymerase will lay down some RNA nucleotides that signal a start point for DNA polymerase, which lays down DNA nucleotides. And when DNA polymerase 3 sees the primer, it attaches and it starts adding nucleotides to the primer. Here's an important detail. Polymerases can only add nucleotides to the 3' end of a DNA chain. Remember how I said DNA is antiparallel? It has a front and a back. Polymerases can only add to the 3' end or the back end of the DNA. This is important because let's say, let me see if I can annotate. Let's say this side is the side of the parent strand is the 5', right? And then this side of the parent strand is the 3'. And this is a 3. We'll just look at this side for now. That means because DNA is antiparallel, the daughter strand, this daughter strand right here will be 3'. Yeah. Because the direction of this strand is this is the 5' end and then this is the 3' end, DNA polymerase can continuously add nucleotides to that 3' end. It will just keep going continuously this way, right? And it can keep adding here because this is the 3' end. However, on the other side, it's a different story. For this daughter strand right here, this is the 5' end. DNA polymerase can't add things. It can't add this way. It can't add this way because it can only add nucleotides to the 3' end of DNA. So what does it do? This side of the DNA is the 3' end. 3'. 3. Yeah. RNA polymerase will continuously lay down RNA primers, and DNA polymerase will continue in these like lagging strand kind of things. Polymerase will make, so what happens is RNA polymerase will lay down a primer, and then DNA polymerase will lay down nucleotides for that section. RNA polymerase will lay down another primer, and then DNA polymerase will run that section again. And then that process keeps going. This is called the lagging strand. The lagging strand, okay. So the lagging strand, these fragments of DNA are called Okazaki fragments. Okazaki fragments right here. And another enzyme called DNA ligase will come in and connect these two bits. If you want to know, DNA polymerase will only DNA polymerase 3 adds nucleotides to the growing end of a strand. However, DNA polymerase 1 replaces RNA primers with DNA. It converts, so these orange bits, this is RNA, but you don't want RNA in your final DNA molecule. So polymerase 1 will come in and replace these. Does that make sense? This is a bit complicated. Anyone have any questions? All right, then I'm gonna get through these. Let me clear these. Weird. Clear. All right. Okay. Enzymes can remove and replace incorrect nucleotides and mismatch repair. So, DNA. I might have to go like 2 minutes over. Apologize for that. DNA polymerases often have a built-in mechanism where what they'll add a nucleotide and they'll check if that nucleotide is correctly paired to the parent strand, right? If it's incorrect, then if it's not correct, the polymerase will like remove that nucleotide and attach and try again, and then it'll keep doing that until it's correct. This is an example of inherent what should I call it? Inherent the polymerase has a self-proofreading function. Another example of repair is our nucleotide excision, DNA excision repair, where if there's a piece of damaged DNA, a nucleus will be attracted to that site, and it will just get rid of, completely cut out that section of DNA, get rid of it. And then a polymerase will come in and remake that section of the DNA by reading the other strand that was undamaged. Remember, DNA is made up of 2 strands. If one strand is broken, you can get rid of that part of the strand and then use the other strand to remake the strand that was broken. Telomeres are non-coding nucleotides that added to the ends of DNA. You should know that telomeres will protect the DNA from being shortened. This is I don't have time to get into this today. Because of how the ligand strands work, every time you replicate your DNA, the DNA molecules get a little bit shorter, right? And because it gets a little bit shorter, if there's a protein coding region at the ends of DNA and it gets shorter, that's bad because you don't want to lose any functional proteins if they because of how tightly regulated our systems are. So telomeres will take the place of protein coding regions at the ends of DNA and protect them from getting degraded. So just remember, this is non-coding and has protective functions. Prevents gene shortening. Cancer cells. This is an inherent protection against cancer because after a certain number of replications, a cell will have to die. A cell will be like greatly limited because its telomeres run out, and because its telomeres run out, the DNA replication will eat into the protein coding regions, and the cell will have non-functional proteins. Cancer, because cancer divides a lot, they consume telomeres faster, and then as a result, sometimes this can protect you from cancer. And chromosomes are not just like a loose spaghetti of DNA in your nucleus. You often have histones, and DNA is often wrapped around histones, condensed around them. If you have 8 histone proteins, and DNA is coiled around those histone proteins, 8 histone proteins and its coil of DNA is called a nucleosome. You can add acetyl groups to histones to make them more you can add acetyl groups to histones to make them more transcribable, which means if you add acetyl groups, then the protein will be expressed more often. If you add methyl groups, then the protein will be expressed less often. This is an example of regulation of transcription, which dictates how much protein you make. Okay, we're done. Thank you guys for coming. Yeah. If you have any questions, you can stay behind, but otherwise, our meeting today is done. Thank you.